Hello, everyone, and Josh. Um, I was asked by one of my rotation interviewers to present some data in a 30-minute prompt, and I was gonna just record everything and see how long everything takes, but maybe you can learn something here. So my background is mainly in biomedical engineering. I took a concentration in bioinformatics in my bachelor's, and I also took a lot of part-time jobs in biotechnology all over the place: gene therapy, laboratory robotics, virology, bioprinting, nanocodings, a lot of like material and computer science automation sort of work. And in the last 3 years, I've been working with the Claiborne lab and really focusing my direction in an immunological angle. And I've really taken to contextualizing the CAR T systems that we do with HIV to tracking multi-omic approaches to T cell differentiation, right? So our lab's bread and butter is flow cytometry, but I've adapted a lot of single cell and bulk RNA sequencing assays to our lab, live cell imaging, and oxygen consumption assays through the Seahorse platform. Now that I am in a cancer biology PhD program, I've been really thinking about what systems and information can I kind of build onto that to really complement this idea of multi-omic approaches to T cell differentiation. So my main body of work definitely regards T cell exhaustion. Why T cell exhaustion? Well, when T cells see antigen, they first become activated and then they become exhausted, and that often includes, an increased rate of proliferation, upregulation of effector function. But under continuous antigen exposures, your T cells will move from activated to dysfunctional, exhausted T cells, where they no longer have the ability to proliferate or undergo effector function. While the Claiborne lab mainly focuses in persistent viral infection, that is HIV, this is also a major challenge in cancer therapies in CAR T or immunotherapies. Our lab likes to model in vivo with a humanized mouse pipeline, that is to say a bone marrow liver thymus humanized mouse, and we're able to model T cell exhaustion in HIV exposed humanized mice. So here we're kind of comparing pre-transfer CAR populations against post-transfer CARs, that is to say 12 weeks of HIV exposure, and you'll see that they have a significantly reduced ability to express CD107A, which is a degranulation marker, and granzyme B, which is another cytotoxic marker. So a lot of the work that I'm very interested in is what exactly is happening in between the state of activated T cell and exhausted T cells, and what drives effector function loss and proliferative function loss, or even the development of that. Well, the first transcriptomic data set that I worked with was an ex vivo experiment where we compared pre-transfer CARs right over here before they're exposed to antigen within the mouse system, and ex vivo CARs, a mouse that was exposed to HIV for 10 weeks. And we see what transcriptomic changes occur during this period in a differential manner. So this is an incredibly rich data set, but I'm going to just glaze over some of the main takeaways. Here you'll see the actual barcodes. We labeled all the pre-transfer CARs in this little red mark, and then the post-transfer CARs as a barcode over here. And you'll see that they actually cluster very distinctly, right? So the pre-transfer CARs will only cluster in this area, but the post-transfer CARs can actually have a similar phenotype here. Overall, the distinction is pretty apparent. Among a lot of differentially expressed genes, some of the most literal takeaways here is that our populations of ex vivo post-transfer CARs are losing effector functions, demonstrating that on a transcriptomic level, we're losing genes, we're downregulating genes in cytotoxicity and proliferation, which has kind of been the groundwork for a lot of interesting things that I wanted to see in other systems. I don't have the time to go over this now, but a lot of my work pertains to contextualizing the physiological functions of a lot of these clusters and using pseudotime like root node analysis to hypothesize over how these cells are changing on a physiological level. And I'll emphasize again that what I'm analyzing in this data set is pseudotime. That's not real time. That's more so imagined time through transcriptomic drift. How would we be able to capture this in a very physiological relevant time course assay? Well, this is exactly the kind of assay that I developed in my lab with Dr. Claiborne, where we developed a chronic stim assay that stimulates the chimeric antigen receptor with a cognate antibody that interacts with our surface receptor, right? And there is a huge benefit when you compare it to in vivo systems where you either take serial or peripheral blood assays on a weekly basis, but that has a very poor CAR yield, or you do serial necropsy, which, eventually kills off your entire study. You're losing more power over time, and there's a lot of heterogeneity between the mice. What you get to do in an in vitro bead stimulation is, because this is a flask, you can just draw a little bit of cells, sample them for your functional assays, and continue to culture it. And we like to think of this as a homogeneous medium, but I understand that it's not fully that way. Now, there has been a ton of validation that went into making this system work, but just as a kind of preliminary demonstration, right, our exhausted T cell model causes a reproducible loss of effector function, right? So these are K562s acting like HIV, and over a 3-week time course, your cells lose the ability to kill cells, which is just exactly what you'd expect in an exhausted T cell. This is the same type of plot, except in a lessened effector to target ratio. And this is saying that by 2 weeks, CARs in lower ratios can kill even less. That is, it's more sensitive to the fall off because the individual CAR needs to kill 3 of K562s to really clear this infection. Next, like I was explaining, our systems are very, very much focused around flow cytometry. So I wanted to demonstrate also that we ran a bunch of different flow panels, and we're able to characterize our cells through intracellular and surface stains. So this is a demonstration of how these in vitro systems are demonstrably losing CD107A, TNF-alpha, IL-2, GM-CSF, all in an intracellular stain. And that'll kind of correlate with transcriptomic shifts that we've noted in the single cell pilot run that I was describing. So yeah, this T cell exhaustion system has been extremely versatile in our ability to interrogate different factors that cause the onset of T cells. We've tested a bunch of different systems or functional readouts for T cell exhaustion. Currently, we're on the experiment 7, which is often spanning 4 to 6 weeks. So there's no shortage of data and labor and reagents that went into these T cell experiments that I've mostly read for myself. Just to briefly gloat here, this has been the body of work that I've been working on in the Claiborne lab for the last 3 years or so. And you'll notice towards the end, some of this went into PAR-seq assay development. I'll talk about that later. And CST01, which is more related to the early activation states and modifying the intracellular co-stimulatory domains of our CARs to see how that affects T cell development. In the same timeline, I spent a lot of work, validating a lot of things within our lab, including, different mitochondrial and surface stains and intracellular stains, and validating the cytotoxicity of different metabolic knockout reagents and stimulus beads, InQ site live cell assays, cell site live cell assays, a bunch of flow cytometry, a bunch of bulk RNA sequencing, which I will get to in the next slide. A great deal of work that I spend is also more or less self-taught analysis of a bunch of these bioinformatic pipelines in the context of bulk and single-cell RNA sequencing, right? So we have that initial 10x run that I was talking about ex vivo from a humanized mouse system, but then we ran some quant-seq and mRNA-seq from our in vitro systems. I did a bunch of QRT-PCRs to match some of our theories. And then more recently, we've been looking into PAR-seq. Or this combinatorial barcoding approach that really reduces the amount of cost that you can put into some of these 10x runs, as well as really improving our agency in the quality of our samples. So I guess that I already introduced it over here, but what PAR-seq is very, put simply, a pipeline wherein you are able to fix and permeabilize cells, bank them in -80, and when you're ready to, you can mass label them. And the most recent kit that I did can barcode up to 48 samples through this combinatorial barcoding system, where basically you label the cells with a unique barcode that is assembled on the benchtop. You pull them back together, and then you use stochastics to assign a unique barcode to every single individual cell through plate manipulation. And not only did I really complete the benchtop work, which is not actually that simple, it takes a little bit of labor and a lot of matrix manipulations. I've also become very familiar with all of the methods to troubleshoot and QC different parts of the pipeline, including staining the nuclear integrity to guarantee that your cells are in a good condition, and looking at the cellular morphology on on a bright field basis, and looking at the tape station and qubit readouts, and finally choosing when to actually prune sublibraries if it's going to consume your reads, and a lot of economical approaches to ensuring that your run is going to look good. So here are the first two applications of the Parsec pipeline that I was working on. Both of them regard the CoStim project, which is to say we are replacing the intracellular domain. Most commonly, these T cells, the second generation CAR T cells, will utilize the CD28 or 41BB intracellular domain, but there's a lot of experimental domains that our lab works with. And we're seeing how stimulating them kind of causes differential transcriptomic developments over the course of activation and exhaustion. So the first one that I've really looked at very carefully, this is a 10,000 cell run, looks at the first week of CARs either exposed to stimulus or not exposed to stimulus, with all these different constructs. We can talk about those if you're interested. And then secondly, we just got this data set in on Monday. CoStim 02 tracks the development of several of these co-stimulator domain variable CARs over the course of a 6-week period. And the data set's super interesting. I wish I had enough time to share that with you as well. So what can I say about the first CoStim run, which is, again, just a 1-week span of activation? So one of my first observations is that there was a variety of samples that we were comparing, but one of the greatest drivers of UMAP topology shift is stimulus versus non-stimulus, which is to say that basically here is a dim plot demonstrating when you separate stimulated CARs and unstimulated CARs, you'll notice that the unstimulated CARs are very much enriched for this left-handed cluster over here, while the stimulated CARs occupy all of these things. And I'll talk about actually what that means later on. Like I was talking about earlier, a lot of my work kind of involves looking at the key marker genes and trying to contextualize it to T-cell physiology, right? So I can talk about this in greater detail if we have time, but basically in this right-handed corner, there's two different activated phenotypes. One that has an upregulation of effector functions, different cytokines and degranulation markers, while the other one suggests more of a proliferative phenotype, KIPs and a lot of biomachinery related to division, cell division. And then on the other hand, there's different quiescent phenotypes and activation phenotypes that lead up to this proliferative and effector phenotype. And if you'll segregate each of the samples by co-stimulatory domain, you'll notice that each of them kind of develops in different sectors or clusters, and if you believe the way that I've assigned some of these phenotypes, it has some very interesting implications. For example, why is OX40, the co-stimulatory domain OX40, causing the cells to be more proliferative than that of like an effector phenotype? There's a lot of very interesting hypotheses that we can follow from this. In terms of a more coherent and testable hypothesis, one of the really interesting genes that I've found to be upregulated in response to different variables, particularly stimulus, is this long intronic non-coding RNA link 00486. And I've kind of referenced this with a paper, the only paper that addresses link 00486 to T-cell physiology. So in our data set, there's about like a 2-fold increase of expression in link 0486. It's expressed everywhere, but it increases in magnitude when you, for example, divide the data set up by stimulated and unstimulated. And you'll see there's an upregulation over here. If you look at different clusters, the ones that I've associated with activated phenotypes, the proliferative and the effector one, there's a noticeably reduced expression of that when compared to the quiescent and the intermediate phenotypes. Furthermore, it looks like certain costim domain or certain car constructs influence the expression of link 486. Why is this cluster, this unstimmed category doesn't seem to be expressing as much link 486 as compared to, for example, CD28? Here I've plotted some really key transcriptomic hits that I've been finding in a lot of my data sets, right? Granzyme B is associated with cytotoxicity. Interleukin-2 receptor is a receiver for the IL-2 cytokine, which will increase proliferative function. And it seems like, with both of these somewhat canonical indicators of effector function and proliferative function, our CARs will express less of it and express more granzyme and IL-2 receptor, and vice versa. Maybe this is an indicator that link 486 is really only upregulated in the case that your cells are not behaving fully functionally as effector cells. In this paper by Wang, they discuss the mechanism of link 486, which again, no one really talks about this, and this is in an EBV system, but what they theorized to be happening here is that link 486 is abrogating this transcription factor, NKRF, which is a repressing factor for NF-kappa beta, which has a ton of downstream T cell effects, right? So when link 486 is upregulated, you'll increase TNF activity, you'll create all these downstream NF-kappa beta pathways, and when link 486 is upregulated, there is a very interesting interaction that happens between SLC-A1, which is a canonical glutamine uptake receptor, and that will kind of supply the T cell with glutamine, which will then lead to glutamine metabolism. So putting together some of this narrative, can link 486, for example, drive glutamine metabolism? And I would be really interested in seeing this outside of a transcriptomic data set, but one of the primary enzymes responsible for glutamine metabolism, glutaminase, is completely downregulated in these clusters that I've dubbed as quiescent, but the active clusters are have a significantly more expression of GLS1. And that has a lot of very interesting effects downstream of everything, because if our T cells are expressing glutamine metabolism, this is mainly explored in cancers, but you can develop an addiction to glutamine. And that has some pretty disastrous effects for cancers when you deprive cancers of glutamine. So maybe this is actually a mechanism of T cell exhaustion. That's it for now for the link 486 story. I'm very interested to see how that how the expression of 486 occurs in a longer term assay. But moving on to another thread that I thought was particularly interesting is that our ICOS construct, that is to say, you take a truncated ICOS that only expresses the intracellular domain and then graft it onto the CAR T cell, only that CAR T variant will upregulate ICOS in response to stimulus. Now, you'll kind of see that this is really only happening in the active clusters. Maybe this is driving some sort of activation phenotype, and you don't really see that much in the quiescent clusters. And just out of curiosity, I also aligned the truncated ICOS transgene, and it seemed like this endogenous, like full transcript ICOS is completely independent of the actual CAR gene expression. Now, just as a proof of concept that this isn't happening in other CARs, I was looking at the 4-1BB expression of the 4-1BB intracellular domain CAR, and to a lesser extent, you actually do see something interesting here, where the 4-1BB CAR and the ICOS CAR will upregulate the full-length 4-1BB transcript in response to antigen, or at least while it's expressing this active-like transcriptional state. And I would believe this as in another study we found on a proteomic level that our CARs will begin to upregulate CD137, this is the same thing as 4-1BB, on a surface proteome level. So this is another very interesting thing, is 4-1BB and ICOS also auto-regulating their own endogenous gene? That would be a very interesting thread to follow. And now I'm going to just cycle through all of the constructs and their, same full-length transcript, right? So you can't find any CD28 upregulation in the CD28 group. You can't find TACI upregulation in the TACI group. I wouldn't believe these as outliers. I could say the same about OX40 over here, OX40 over here. Yeah, so what exactly can you take away from here? Well, ICOS is an inducible co-stimulatory domain that acts similarly to CD28, but it's almost never the case that they operate identically. In fact, if nothing else, you know, they appear to cluster somewhat similarly. There's not as much as a apparent polarization as is in TACI and OX40. But I think this is just a really neat hook that doesn't necessarily make sense that I'm really excited to see in future scRNA, single-cell RNA datasets. So, like I said, we just received the second run. That is a 100,000 cell single-cell run. I'm very excited because the quality of these samples look excellent. I put a lot of work into improving the process, pruning some libraries that didn't look super good, and it seems to have bared a very, very interesting data set that I'd love to dig into soon. I think that it's also very interesting that we've included time domain now. We have time points at week 0, 1, 4, and 6, so we can really see what's happening by this 6-week, presumably exhausted phenotype, as the cells have seen antigen over a 6-week period. Some things we might be looking for, can we corroborate some of these Link486 and ICOS data in week 1? And what's happening in a longer term? Does it have any consequence when the initial cells are expressing first the Link486, and does it continue to express? Does it have any continued effects on effector function development or even loss thereof? And also, we ran a bunch of matched ICS data sets on this CoStim2, so I'd be very interested to see if we could match some of the transcriptomics with some of our flow data, some of our proliferation data, some of our killing data. That would be very interesting. Here's a brief screenshot of what the CoStim02 looks like. I don't have a lot to say about this, except there's some range of variability between sublibraries. If nothing else, this blue bar is actually an indicator that maybe this library was not worth sequencing, and that's what I mean by pruning sublibraries. There are certain sublibraries that just don't necessarily have the same quality as others, which is really nice that you do them in replicates, not in a giant pool. It's that you're able to do QCs on them and say, hey, this might not be worth the cost of sequencing, because at the end of the day, that's really what drives costs. Overall, it seems like this data set looks really, really good. There's a ton of unique transcripts per cell, and I'm pretty excited to dig into that. Yeah, so I tried to keep this focused towards the parseq pipeline, as I thought that you guys might be more interested in that one, but I have a lot of text data sets and a lot of very interesting conclusions that I put together from trying to integrate several multi-omic approaches to our T-cell exhaustion projects. Now, some things that I just briefly wanted to mention that I'm very interested in is this mitochondrial landscape restructuring when it comes to activation and dysfunction. Each of them have a very distinct potentiation and biomass, and it's another very interesting thing to integrate on a transcriptomic proteomic level. Another thing is surface phenotypes when it comes to staining a bunch of flow. Basically, you'll notice that a lot of our T cells stop acting like canonical T cells as we would expect, and I'd love to dig into what exactly is happening once these inhibitory receptors see the ligands that they really shouldn't be, but these T cells are kind of becoming the exception. Some T cells recover function. I'd love to look at on a single cell transcriptomic level what's actually driving that. And lastly, there's a ton of evidence that's suggesting to me that a lot of T cell exhaustion occurs on a epigenetic and transcriptional regulation landscape, which I'd love to learn about. But anyway, there's a ton of supplemental data. I hope we've saved enough time for questions, and I will take them now. Thank you.